Okay, so first question, we have a winner, the histone code. So we're talking about epigenetics and the histone code. When we're talking about methylation and acetylation, how do you know if we're talking about methylation or acetylation of the DNA or of the histone, the protein? I'd have to tell you. So in genetics, we can do different treatments if we take some organism's cells and we want to know, is there acetylation on a histone or acetylation, which doesn't happen on DNA, but methylation, say, on histone versus DNA. Use different extraction techniques. So you separate the DNA from the protein, and then you can assess whether or not the acetyl marks are on the DNA or on the protein. So I would have to tell you which one I was showing you, but experimentally we determine it by doing extractions. Look at the proteins and histones and what modifications they have versus the DNA. Is that a sufficient answer, or do you want more information?
Well, like, when we were doing like a practice problem, we were like, which mutation turned off the X1 or the X2, and like someone said they would use trimethylation, or whatever. We said, which one is it, DNA or lysosomal?
Right, so the point I was making, one of the practice questions had something to do with how do you turn on or turn off gene expression? Is it methylation of DNA or is it methylation of histones? And the important point there was to underscore the idea that normally methylation marks on DNA or histones cause gene silencing, inactivation. And normally acetylation causes, which is only histones, DNA doesn't get acetylated. Histone acetylation usually leads to activation. The general patterns. There are lots of exceptions to that, but that's sort of a general trend. Methylation, genes turned off. Acetylation, genes turned on. Sir?
I was just wondering, how are we going to be tested on more conceptual concepts like GMOs and stuff like that?
So how would I test you on something like a GMO? That is an excellent question. Yeah, it's true. What do you think? I didn't ask you to make a study guide, but what sort of a question do you think I would ask about GMOs? I haven't written the exam yet, so you could actually maybe help me think of a question to ask about GMOs. I could provide a list of scenarios, short scenarios, and ask you to pick one that you think is genetically modified and explain, what is your definition of GMO and why do you think it applies most to this scenario organism, say, compared to the other three. That would be one way I might do it. And how many points is this exam worth? 20, I think. Which is unfortunate because I wish I could ask way more than 20 questions, but then they'd be worth partial, like half-point questions and stuff. So I will not be able to, the point is, I will not be able to cover all of the material from the semester. It'll be 20 questions or less, hopefully much less, which means that you're going to study a lot to try to cover, because it's a cumulative exam, all of the topics we've talked about, but not all of them will show up on the exam, obviously. And then you will have, as I said, the full 2 hours to take the exam. It'll probably be a midterm and a half worth of information, or worth of questions. So you'll have a little bit more time on average per question, I think, to take the final. It'll have exactly the same format, open note, etc., open internet, distributed and collected by Google Classroom. So we've talked about methylation and acetylation. We've talked about the histone code a little bit. What other topics, or what amount of depth would you like to go into? Yeah.
Can you explain more about the gal pathway?
Okay, so the gal pathway. So this was the example of eukaryotic gene expression regulation. We talked about the lac operon in bacteria, and we talked about the gal pathway. All right. And that was a eukaryotic yeast, but eukaryotic example. So what would you like to know about it? Should we just review how this works? Okay, so yeah, we're talking about genetic pathway. You've got inhibition going on. In fact, didn't we have an assignment about drawing the gal pathway in a genetic pathway? Okay, so what's the gene that needs to be transcribed for galactose utilization? It's gal1, right? I think. It's an enzyme that needs to be produced to utilize galactose. What's the protein that will turn on transcription of galactose 1? What's the transcription factor, the regulatory protein that comes and binds an enhancer? That's, say it again? Gal4. So we can have the protein gal4, which will bind to enhancer, and that positively regulates arrowhead, positively regulates gal1. So what happens next? So that's on. How do we turn it off? How do you keep gal4 from turning on lac1, or gal1, sorry. How do you keep gal4 from turning on gal1 when there's no galactose around? What do you think? What's the protein that represses gal4? Something can turn it off. This is gal80. So gal80 can prevent gal4 from activating gal1. Where does galactose fit into this whole thing? Somehow, like in the lac operon case, there has to be some way for galactose to play a role here. The sugar itself has to give information to these proteins about whether or not it's available to be digested. How does that happen? Who regulates gal80? So galactose binds to you've got a protein that regulates a protein that regulates a protein. Where does galactose interact with this system? It's with gal3. So when galactose binds to gal3, those things remove gal80 from the equation. And without gal80, so in the presence of galactose, the protein gal3 is now able to repress gal80. You have no gal80, then you have gal4 active, it turns on gal1. So I've drawn the arrows here like the genetic pathway. It's a series of repressions followed by one activation step. So something regulates something that regulates something negatively. Ultimately, gal4 is the thing that turns on expression of gal1. What would happen if you had a mutation of gal3 so that it couldn't bind galactose? What effect would that have on this system? So galactose is present, gal3 can't bind to it. So is gal80 going to be active or inactive? Active, right. So if gal3 is not able to bind galactose, it can't do this step. So then gal80 would be active, gal4 would be repressed, and there would be no gal1 produced. So that would be a bad situation for the organism because galactose is there, but if gal3 can't bind to it, then gal1 isn't turned on, the organism can't use the sugar source for energy. So that's one sort of way I might ask you to interpret or manipulate or understand the sense of the pathway. It's genetics, it's about mutants. Questions, clarifications, additional thoughts? Yeah.
Can you do the genetic pathway for the worm sex determination?
Nematode sex determination pathway. So let's see. First, we have to talk about what are the chromosomes that cause sex in a nematode? So what are the two different chromosomal constitutions of the two different sexes? We have hermaphrodites and we've got males. So males are XO. Hermaphrodites have two X chromosomes. Now this is, if I remember correctly, this is a series of repression events. So something prevents something, prevents something, prevents something. Does anybody remember the first protein that's involved? So ultimately it has to do with whether or not the right end of the pathway, the outcome is whether or not hermaphrodite-specific genes get turned on. Okay. So where does TRA, that's upstream, or that's the left end or the right end of the pathway? Where does TRA1 fit? Is it TRA1 first and then the FEMs? So TRA1 represses a protein called FEM, which represses a protein called TRA2. And then what's the relationship between TRA2 and the hermaphrodite-specific genes? Does TRA2 activate the hermaphrodite-specific genes? Always got to have this in their notes. Yeah? So TRA2 is, that's a solid arrow or an arrowhead. Right. So we've got a series of repression events. I'll draw these arrows in black so they stand out a little bit more. So TRA1 represses the Fem proteins, which repress TRA2, and TRA2 is an activator of hermaphrodite-specific genes. So I guess the last question then is, how does the sex chromosome constitution impact TRA1? So if you're a hermaphrodite, let's figure it out. You don't even need to look in your notes. We can figure it out from here. So if you're hermaphrodite, what's the outcome that is going to happen? If you're XX, what sex are you going to be? You're going to have your hermaphrodite-specific genes turned on or turned off? Come on. Turned on. Yep. So above the black line, so if those genes are going to be turned on, those genes are going to be transcribed to produce an XX hermaphrodite. Then what do we want TRA2 to be doing? Do we want TRA2 to be active or inactive? This one right here. So TRA2 turns on hermaphrodite-specific genes. So we want TRA2 to be present or absent in a hermaphrodite. Present. Right, so if you've got TRA2 turned on, it's a transcription factor. It's going to go bind to genes and turn on the hermaphrodite-specific genes. So TRA2 has to be present. So if TRA2 is around inside the nucleus to make a hermaphrodite, then if Fems repress TRA2 in a hermaphrodite, are the Fems present or absent, active or inactive? They're going to be inactive. So if you don't have the Fems around, then they can't repress TRA2, so TRA2 is active. What does that tell us about TRA1? TRA1's normal job is to repress the Fems, so it has to be around. It's the thing that turns off the Fem genes, the Fem proteins. So what does that tell us about the interaction between the X chromosomes and TRA1? If you're a hermaphrodite, this says to us so far, if you're a hermaphrodite and you want your hermaphrodite-specific genes turned on to develop you as a hermaphrodite, you need to have TRA1. So do we put a blunt arrow or an arrowhead between you've got two X chromosomes and TRA1? Yeah, arrowhead. Two X chromosomes, TRA1 gets turned on. It represses the Fem, so they're absent. In their absence, TRA2 is turned on. It turns on the hermaphrodite-specific genes. And then without two X chromosomes, the XO sex, the males, repress TRA1 instead of activate it, and then the hermaphrodite-specific genes ultimately are turned off. And that's because if without TRA1, the Fems are present, TRA2 is inactive, so you don't get hermaphrodite-specific transcription. What sex would a worm develop as if you had a worm that was a TRA1 mutant? So it didn't have that gene. Let's say that TRA1 gene didn't exist, so it couldn't make the protein. What sex would it be? This is going to look an awful lot like this bottom pathway here. When you don't have TRA1, right here. If you've got a mutant that doesn't have TRA1, then you're going to have the FEM's active. So TRA2 is going to be repressed. So which sex does that make? A TRA mutant. Yeah. So that's, in fact, why TRA was named TRA. It stands for transformer, not the kind that is more than meets the eye, but rather it transforms what would be a hermaphrodite into a male. And the FEM genes are named FEM genes because when they have mutations in those genes, they feminize hermaphrodites. That is, they turn hermaphrodites into the opposite sex. Again, an example of a question I might ask related to that topic.
So on the final, it's going to be partially this stuff that we covered after the last test, and then just everything else.
It will be a comprehensive final. So yes, there will be a mix of material that was covered on exams 1, 2, and 3, and 4. Were there 4 exams? And everything that's covered since the last exam, whichever number it was. And once I actually write the exam, I'll do my usual Blackboard post giving a little bit of the statistics about the exam, and I'll try to give you an idea of what balance between old material and yet untested material will be on the final. So what did we cover since the last exam? GMOs, epigenetics, sex determination. What else? Oh, right, and gene expression regulation. We've already talked about epigenetics, gene expression regulation, GMOs, sex determination. We've done one example of each of those 4 topics so far. Others? All right. So what was the concept of genetic linkage? Why did we need to know about probability? So what do you need to know about genes to know if they're linked or not? What's the essential piece of information you need? How do you tell if 2 genes are linked? Okay, so their distance. Which distance? Yeah, genetic distance. You want to know how many centimorgans they are apart. So what's the critical value here? $50. So what does it tell us if 2 genes are 50 centimorgans apart? Are they linked or are they unlinked? Right, this is unfortunate. They're either unlinked or they're at the opposite ends of the same chromosome. which is where two genes could be linked physically. They're on the same chromosome, but they act like they're unlinked. So what we would use genetics, or what we use statistics for, is to ask, is there a significant difference between the observed recombination rate and 50? So the null hypothesis, our expectation we start with is that genes are unlinked. They're 50 centimorgans apart. And what we're looking for is evidence that the genes are closer together genetically than that. What sort of a cross design do we use to assess genetic distance? Let's say we've got this sort of a cross. There's two parental P0 individuals. They create an F1 dihybrid. Who do we cross that individual to tell whether or not genes A and B are linked? What's the genotype here? Either the genotype or the name of the type of genotype. Tester, right. Which is what? All lowercase, right. So homozygous, lowercase. And those individuals can produce then 4 different types of offspring. You get big A's with big A's, well, more than 4, but for this point, you get big A's with little a's. You can get little a's with little a's. And for each of those types, you can either have a big B with a little b or a little b with a little b. Making these 4 possible genotypes. So it's the ratio of these 4 different types of F2 genotypes that tells us something about whether or not these genes are linked. What's the parental haplotype? That's right. Now we have to figure out, we know that a little a and a little b came from the tester parent. So we know the origin of those little a's and little b's. So it's something about the ratio of the leftover genotypes, I'm circling in blue. These haplotypes came from the dihybrid parent. So we want to know, are there a lot of parental types or are there a lot of recombinant types? What's the ratio? That's how we calculate genetic distance. So is capital A with a capital B, is that a parental type or is that a recombinant type in this example? How do we know? How do we define what a parental type, haplotype looks like? We have to look back here at the parents and the genotypes, or the haplotypes rather, in their gametes. So these parents would make capital A, little b gametes and little a, capital B gametes. So those define our parental types. So So that would be this one and that one. And then we'd have two recombinant types. So anytime we see an F2 individual that has a big A with a big B or a little a with a little b, those are recombinants. So what if I tell you we've got 41 of those and 37 of those, and 12 little a, little b's, and 10 big A, big B's. Just making numbers up. Hopefully it all works out in the end. Looks like it. So that's 100 total offspring that we're looking at. We've got 100 F2s, we've got their genotypes. The question then I'll ask you on the exam is, are these genes linked or unlinked? So I'd probably start with this table. I'd say, here are some F2 genotypes, here's the cross that was used to produce them, and then these are the numbers of the 4 different genotypes you observed. 10, 41, 37, and 12. Are the genes linked or not? What do you do next? How do you calculate genetic distance? Centimorgans is 100 times the recombination fraction. How do you calculate recombination fraction? So the number of recombinants, 10 plus 12, divided by the total number of individuals, which in this case is 100. So what's the genetic distance between A and B? In centimorgans. 22 over 100 times 100. So 22. So are these genes linked or unlinked? Wait, less than 50 centimorgans. But where chi-square testing comes in, and this is now the second part of the question, why do we use statistics in this analysis? We need to know if we've looked at enough individuals. Is 100 enough individuals, F2s, to look at to know for sure that 22 centimorgans is absolutely different than 50 centimorgans? If you look at too small a number, if you sample too few F2s, you might by chance calculate 22 centimorgans even when genes are unlinked. So how do we do the chi-square analysis here? We've got observed values. That sum to 100. What would we have expected to see? How many of each of these 4 different genotypes would we have expected to see under the assumption that they're unlinked? Or that they assort independently? Instead of 10, 12, 37, 41, what numbers would we have seen? Mendel's laws are working. Independent assortment is occurring. Yeah, you'd see a 1 to 1 to 1 to 1, 25 of each of those 4 different genotypes, so that you get the same total number of observations. So to be explicit, that's these genotypes. So from now, hopefully this will all come back pretty quickly. To do the chi-square test, you take the observed value, you subtract the expected value, square it, divide by the expected value. That's observed minus expected squared, divided by expected. And you do that for each of these 4 different categories, 4 different genotypes, and you sum all 4 of those numbers together. So the chi-squared test statistic value, the sum of those 4 numbers, when you calculate them all, 31.76. Linked or unlinked? What do we have to do next to really answer the question? We have to have 2 things to look up on a chi-square table. We've got the chi-square test statistic value, and we need to know degrees of freedom, which in this case is 3. It's the number of genotypes, or the number of haplotypes, which in this case is 4. Big A with big B, big A with little b, little a with big B, little a with little b, minus 1. So we've got our chi-square value, and we've got our degrees of freedom. 31.76, 3 degrees of freedom. Oh, look, I've got a chi-square table. So we look on this line where we've got degrees of freedom is 3, and we're going to read across until we find our chi-square value. So left to right, where's 11.76? Somewhere in between 11.3 and 12.8. Wait, 31. Sorry, that's way over there. So what's the p-value? These are all the p-values, the probability that the null hypothesis is correct. What was our null hypothesis again? Our expectation? These genes are unlinked. So the probability that the genes are unlinked is going down the bigger the chi-square value gets. So we're at, let's see. A p-value of 5.8 times 10 to the minus 7. Really small probability that these genes are unlinked. So your conclusion would be, if the genes are not unlinked, they're linked. If you want to say it formally, you'd say the genes are statistically significantly linked. And in your case, because you don't necessarily have Microsoft Excel running to calculate the exact p-value on an exam, you would just say the p-value is less than that smallest value. It's less than 0.005, but that leads you to the same conclusion. If the p-value is less than 0.05, you reject the null hypothesis. One example of a linkage question in chi-square analysis. 5 more minutes. Aside from getting out of your way, what's the next best way I can help you at this point? Going to do a quick pedigree analysis. Off. Make one up and see what happens. I have no idea what I'm doing right now. I'm just going to write some symbols. Well, and this is what it's like in the real world of genetics. You never know who's going to walk through the door and show you their pedigree, so you never know what it's going to look like. So you might as well just make up something. So I'll be just as surprised as you are when we figure out the answer. Hopefully there is an answer. There must be an answer, mustn't there? So if somebody comes to you and they have this pedigree, which I totally just made up, I didn't even try to make a particular pattern. How do we start? What's the first thing to look for? Is it skipping a generation? So in this case, step one, not skipping a generation, so let's start assuming it's dominant. Okay, so we've got two types of dominance. We've got autosomal dominance and X-linked dominance. So the next step is, can we rule one of those out? So let's start with, could it be an X-linked dominant trait? So what's true about X-linked dominant traits? If mom, what's mom's genotype? The affected mom, that is. So let me number some individuals. So individual 2, what's her genotype? If this is X-linked dominant, it's our assumption, she could either have one bad X chromosome, X minus over X plus, or she could be homozygous for a mutation. Either of those genotypes would give her the disease, trait, whatever. Yeah?
So, I just want to make sure I'm clear. So there's the 2 sets of parents producing the 5 kids?
Yeah, isn't that weird? So, see, now we've got somebody who's actually paying attention. Now, I'm not saying none of you are paying attention, but okay. Voila. Case problem solved? Now you've learned an important lesson. Never ask me to make something up on the fly. Is that easier? Okay. I'm not going to ask anybody to do anything that's not actually biologically feasible on the exam. So this should make it easier. Thank you very much. So which of these 2 genotypes could she be? We don't know yet. Maybe we can figure this out. So mom passes her X chromosome to sons or to daughters? Both. So could this be X-linked dominant? So mom could pass an X minus to a daughter and an X minus to a son. And then the daughter would get, let's assume dad, if this is dominant, what's dad's genotype if this is X-linked dominant? He's got a Y chromosome. What's his X? Plus or minus? It's got to be plus, because if he was X minus, he had the mutation, he'd show the trait, and he doesn't. So that means dad is passing an X plus to his daughter, and what did he contribute to his affected son? His Y chromosome. So far, this pedigree is consistent with the possibility of it being an X-linked dominant trait. And all of the other kids could get an X plus from dad and an X plus from their mom, assuming that the mom is heterozygous. So we're going to assume that those genotypes have been circled are the parents' genotypes. And bottom line, I know it's time to go. Bottom line for this analysis is if you can prove that X-linked dominance is possible, then it's just as likely, if not more likely, that autosomal dominant is also possible. So if in this case we've said it's possible that it's X dominant, then you would say it's also possible that it's autosomal dominant. Good luck studying. Have an excellent weekend when it gets here, and I'll see you at the exam.